Welcome to another deep dive. Today we're going microscopic, like really tiny, to explore oocytes.
Oocytes.
Yeah, the cells that can create new life.
Yes.
And we're going to look at how they manage to stay so healthy.
For decades.
For decades.
Crazy.
Right?
Just incredible.
And the way they do it, well, we're unpacking some excerpts from a research paper for this.
Okay.
It's called Oocytes Maintain ROS-Free Mitochondrial Metabolism by Suppressing Complex I.
Catchy title.
It is. And it turns out their survival strategy is really something else.
Definitely not what you'd expect.
No, not at all. It's not like anything else we see in the animal kingdom, at least as far as we know.
Yeah, it's really remarkable. You see, oocytes face a unique challenge. Unlike most cells in our bodies, they can remain, as we said, viable for decades. But during that time, they're constantly exposed to reactive oxygen species.
ROS.
ROS, right? And those are the harmful byproducts of cellular metabolism.
Yeah, ROS, it's kind of like rust.
Rust.
Yeah, like rust that forms on a car.
Ah, okay, I see.
So oxygen is essential, but using it can lead to these unwanted side effects, right?
Right, right.
Too much rust, or in this case ROS, can really mess things up.
In a cell, definitely. Yeah. For oocytes, keeping ROS under control is critical. You know, they need to stay in pristine condition if they're going to create a healthy organism.
So how do they do it?
Well, that's where it gets really interesting. This research, it reveals that oocytes have a way of essentially switching off.
Switching off?
Yeah, switching off a part of their mitochondrial electron transport chain.
Okay, so which part are we talking about here?
Complex I.
So they basically switch off complex I.
Right.
And that's a surprising finding.
Very surprising.
It really challenges how we think about how cells function.
Exactly.
So most cells, they rely on complex I, right?
They do, yeah.
And that's a key part of the energy production line in the mitochondria.
Like the first domino.
The first domino.
Yeah, it starts this chain reaction.
That generates energy.
For the cell, yep.
Okay, so complex I is important, but oocytes are surviving without it.
They are.
How is that even possible?
So they've developed a workaround. They compensate for the loss of complex I by keeping other parts of their energy production system active.
And what system is that?
The OXPHUS system.
OXPHUS. Okay, so they keep that running.
They do. They're basically rerouting the energy production process to bypass complex I altogether.
Wow. That's some serious cellular ingenuity. And they didn't just find this in human oocytes.
Nope.
The researchers saw the same thing in frog oocytes.
Frog oocytes, that's right.
So this suggests that this strategy has been conserved throughout evolution.
Yeah, the fact that this mechanism is found in such diverse species points to its importance for, oocyte survival and also their incredibly long lifespan.
Yeah, that makes sense. Let's talk about the evidence that led to this discovery. What kind of experiments did the researchers do to reach these conclusions?
Well, they used several techniques, both on human and frog oocytes. They combined live cell imaging, sensitivity tests, and respiration measurements. And from all that, they were able to demonstrate that early oocytes had no detectable ROS. And it wasn't because they were just really good at cleaning it up.
Oh.
They actually prevent ROS formation in the first place.
So they stop it before it even starts.
Exactly.
Fascinating.
They also confirmed that the oocytes still had functioning mitochondria, just operating at a lower level.
So they're not sitting down their powerhouses entirely. They're just sort of dimming the lights a bit.
That's a great way to put it.
But how did they prove that complex I was truly absent? I mean, how do you prove something's not there?
Yeah, that's a good question. They used a technique called proteomics.
Okay.
And that allowed them to analyze the proteins present in the oocytes. And what they found was a significant depletion of the protein subunits that make up complex I.
Okay, so the building blocks were missing.
Right. These building blocks were so scarce that complex I couldn't even assemble properly.
So it's like they're missing some of the key ingredients for complex I.
Exactly. And to be absolutely sure, they directly measured complex I activity, and they found it to be completely absent in early oocytes.
So no complex I activity. Got it.
They also looked at the levels of a molecule called FMN, which is, a vital component of complex I.
Right.
And found it was practically non-existent in these oocytes.
Okay, so we've got missing proteins, no activity, and depleted FMN. It seems pretty clear complex I is just not present in early oocytes.
Right. And this is a real paradigm shift for us.
How so?
Well, in our understanding of cellular biology, before this, we didn't think any animal cell with functioning mitochondria could exist without complex I.
So oocytes are rewriting the textbook.
They really are. So we've established that oocytes can function without complex I.
Yeah.
Which is pretty remarkable in itself.
Definitely.
But there's another fascinating aspect to this whole thing.
Okay.
A kind of cellular quality control system that comes into play when complex I is absent.
Interesting, like a backup system.
You could think of it that way.
Okay.
It's called the mitochondrial unfolded protein response.
Wow, that's a mouthful.
It is, yeah, UPUMP for short.
UPUMP. Okay, I'll try to remember that. So what exactly is it? What does it do?
Well, think of it like a cellular mechanic.
A mechanic.
Yeah, one that gets called in when things aren't running smoothly.
Okay.
Within the mitochondria, you see the mitochondria, they're very sensitive to imbalances.
Imbalances.
Yeah, in their protein composition.
Okay.
When something is off, like when complex hair is missing,
Right.
the UPM, it kicks into gear
Okay.
to help repair or remove damaged proteins.
So it's like a cleanup crew.
Exactly. It helps to store that balance.
So it's like a built-in stress relief mechanism for the mitochondria.
You got it.
And the research shows that the UPMP is highly active in these early oocytes, right?
That's right. It's likely a direct response to the absence of complex I.
Okay, makes sense.
And here's where it gets even more interesting, UPMP activation.
Hmm?
It's been linked to increased lifespan.
Wait, really?
Yeah, in other organisms.
Hold on, are you saying that by not having complex
Yeah.
I, the oocytes activate this UPRMD,
Right.
which could actually help them live longer?
That's the idea. It's like they've stumbled upon a longevity hack.
A longevity hack.
Yeah, at the cellular level.
Wow.
By suppressing complex I, they unintentionally triggered this UPRM response.
Okay.
And that may contribute to their extended lifespan.
That's incredible. It seems counterintuitive
It does, doesn't it?
that missing a key component would lead to a longer lifespan.
Yeah, but I guess that's the beauty of evolution, right?
Yeah.
Finding these unexpected solutions to complex problems.
Absolutely. And we have to remember, oocytes aren't just sitting around doing nothing.
Right.
They are metabolically active.
Even in their dormant state.
Right, they still need
To
energy,
carry out essential functions.
but they do it in a way that minimizes ROS production.
Exactly.
So they've managed to strike a balance between their energy needs and longevity.
Precisely. They've optimized their metabolism to ensure both survival and functionality.
Over an incredibly long period.
A truly remarkable adaptation.
It really is. And this whole complex I bypass, it has implications for how we understand mitochondrial diseases, doesn't it?
Absolutely. The fact that oocytes function perfectly well without complex I, it helps explain why some people with inherited complex I deficiencies don't have fertility issues.
Because their oocytes are naturally equipped to handle it.
Exactly. Their oocytes have evolved this unique strategy
That makes them less vulnerable.
to the negative effects of these specific mitochondrial mutations.
Okay, so let's recap. Oocytes have this remarkable ability to suppress complex I,
Right.
which helps them avoid ROS damage,
Yes.
and potentially even extends their lifespan through the activation of UPRMD,
It's a fascinating example of how cells can adapt and thrive.
even without what we thought were essential components.
Yeah, and I'm sure our listeners are wondering
Yeah.
what this means for the rest of us.
What does this mean for us? Could we use this knowledge to benefit human health? Okay, so let's get to the part of it. We've been talking about how oocytes stay healthy for so long. And it boils down to this ability to bypass complex,
Yeah.
which most cells need for energy production. But now the question is,
Yeah, the big question.
could we apply this knowledge to other cells?
It's an exciting possibility.
Could we revolutionize how we approach disease and aging?
Maybe. Think about it. All those diseases,
Uh-huh.
they're linked to damage from those reactive oxygen species.
The cellular rust.
Exactly. And even aging itself.
Aging?
It's partly a story of accumulating that ROS damage.
So what if
we could switch off complex I
could oocytes do
in other cells? Yeah.
to protect them from this damage?
That's the idea.
That's a fascinating idea. So we could shield healthy cells
Attention.
from things like the side effects of cancer treatments,
Right.
or even slow down aging.
It's a possibility.
But wouldn't messing with complex I in other cells
Yeah.
have downsides?
Of course, you're right. I mean, it's usually there for a reason.
Right.
Most cells rely on it
Heavily
heavily, yeah, for their energy production.
for a long perspective.
Extended periods, yeah, it could have unintended consequences.
And oocytes are unique, right?
They are.
They can survive in low power mode.
Decades. Yeah.
Might not be so adaptable.
Exactly.
So it's not a simple solution.
Yeah.
Just flip a switch. There's a balance. We need to figure out how to,
How to inhibit it.
inhibit complex I
Well
temporarily
enough,
to protect the cells.
but without causing harm.
Right. And that raises a question.
Yeah, a big one.
What makes oocytes so resistant
To those downsides.
of complex I suppression?
Right. Is it something specific about their energy setup?
Or some other safeguard?
We need to figure that out.
If we could, it would be amazing. We could tailor the complex I bypass.
To different cell types?
Yeah, fine-tune it for maximum benefit. Wow. It's like oocytes have given us
A glimpse.
into a whole new world
It's possibility.
for cellular manipulation. Who knew that studying these tiny cells could lead to,
Such a groundbreaking insights.
with the potential to change human health.
It's amazing, isn't it?
It really is. It's a testament to, well,
Scientific curiosity.
yeah, and exploration. Sometimes the most unexpected discoveries lead to the most profound advancements.
Absolutely.
That's why we love these deep dives, uncovering these hidden wonders of science, exploring how they might shape our future.
It's fascinating stuff.
It is. Thank you for joining us on this incredible journey. We hope you learned something new and exciting, and maybe even a little mind-blowing. Until next time, keep exploring and stay curious.